our second talk of the afternoon, entitled, I'll speak slowly.
Okay.
Okay. She's, this is some of her work with Dr. Gianella, who couldn't be here today, so she's presenting some of their joint work, and the title is Compartmentalized HIV Rebound in the Central Nervous System After Interruption of Antiretroviral Therapy.
Okay. So this is another study from our group, as Mary said. So different research groups, including ours, have been shown that likely there is a compartmentalized HIV reservoir in the central nervous system when compared to the periphery. And this compartmentalized HIV DNA reservoir may be established in the central nervous system very early, and perhaps regardless of early start of antiretroviral therapy, as the data that we just showed suggests. So, saying that, we had important questions that lead to this study. So, our main question was, is there a replicative competent HIV DNA reservoir in the central nervous system that contributes for the viral rebound after interruption of antiretroviral therapy? And which factors would be associated with compartmentalization of these viral populations between blood and cerebrospinal fluid? So for this study, we selected retrospectively paired blood and cerebrospinal fluid samples from 14 chronically HIV-infected individuals that undergone voluntarily to ART interruption. And we analyzed the first two available time points after the viral rebound. So in this table, we have the summary of the baseline characteristics of this cohort. And again, mostly these participants, they were Caucasian males with median of age of 41 years. And they presented a median time to HIV rebound in blood of 19 days and 30 days in CSF. And the time from the virus rebound to the sampling was 24 days for blood and 14 days in CSF. So as we already sort of saw here in this table, but here it's better in this representative graph from one of our subjects, we can see is, and we saw this overall across the subjects, is that after ART interruption, the HIV rebound in cerebrospinal fluid had a delay in comparison to blood. And this delay had a median of 10 days. So for these subjects, we also generated ultra-deep sequences for 3 genetic regions of HIV, envelope, GAG, and RT, that were amplified from cell-free HIV RNA in blood and CSF, and we used, again, the 454 platform. We also measured the levels of immunoactivation by CD163, IL-6, TNF-alpha, and MCP1 in blood and CSF. So here is the table summarizing the compartmentalization status in the first time point for the 3 regions across our 14 subjects. And what we can see is that almost all of them presented significant signal of compartmentalization in at least one of the genes. 8 of them presented compartmentalization in all genes available, with sequences available. And interestingly, we saw that half of these patients already presented signal of compartmentalization with a time below 2 weeks after the time to rebound, the time of viral rebound. So again, we constructed phylogenetic trees with sequences from obtained from blood plasma, as represented by the circles, and CSF represented by the squares. And here we have in orange the first time point, and in green the second time point. And again, we were able to see that this population that, this viral population that rebound after interruption of antiretroviral therapy, they have sequences that, again, they were CSF-only sequences that were not present in blood, showing that this viral rebound is also compartmentalized. And we observed these CSF-only sequences in 13 of our 14 patients. So finally, we also did exploratory analysis looking if levels of inflammation could be related with levels of compartmentalization. So what we saw from all the predictors that we evaluated as viral load, CD4 counts, and other predictors, we observed that high levels of CD163 ratio blood CSF was associated with low FST, which is a score that as the result of compartmentalization is expressed. So it means that increased inflammation was associated with absence of virus compartmentalization. So as conclusions, in most of participants, HIV RNA rebound occurs independently in blood and in CSF after ART interruption. And this compartmentalization persisted, sorry, I forgot to say that, in all 14 participants, the subsequent time points were also showed also compartmentalization. So compartmentalization persists over time. And increasing inflammation was associated with lack of compartmentalization between blood and CSF in our cohort. So again, I would like to thank you, the participants of this study, all our collaborators, and you for the attention.
Any questions?
Do you know which drugs are in this ART regimen?
These are, sorry, I don't remember now from the top of my mind, but I can check and tell you later.
Do you think it was the inflammation that allowed the trafficking to occur, or that the
Most
trafficking
likely, yeah.
it elicited the inflammation?
Well, it can be actually a combination of different events. So, but what we believe is that inflammation is associated with lack of compartmentalization, thinking that inflammation could allow migration of between the blood-brain barrier and blood. But what comes first, it's the egg or the chicken. Perhaps the things are happening together and they might be multifactorial.